I would like to thank the organizers for inviting me here to present some of our work. A lot of the work has been sort of done in collaboration with my former lab, with the Kershaw group in King's College London. A lot of the talk will be actually about RNA toxicities, about RNA foci and sequestration of RBPs. Yeah, so basically there's not a lot of point in introducing here, but the repeat is in the first intron of C9orf72, and the intronic localization of the repeat strongly suggests that the mechanism is involving this RNA species. And basically one of the big questions that awaits us all is that down the line this is a TDP43 proteinopathy. The region, the repeats actually allow very complex DNA and RNA structures to arise. There are possible structures. One is the G quadruplex structure for the DNA. Then there is I motifs, which are structures which can be formed by poly C or C stretches. This hasn't been shown, obviously, for G4C2 repeat, but there are indications that DNA can, stretches of Cs in DNA can form I motifs. There was recently a publication on the formation of R loops, so the heteroduplex of DNA and RNA. And then when we have translations which transcriptions which can happen from sense and antisense direction, we can have G quadruplexes forming from the RNA, and these RNA may also form hairpin loops. I motifs is a bit of a question because RNA hasn't been shown yet to form I motifs, but again, there is a possibility of that, and the stem loops from the antisense transcript, and then obviously there's the possibility of these stem loops initiating RAN translation. So one of the things that one of the studies that we did, we looked at the DNA of G quadruplex, the G4C2, and like it's been shown by Adrian, for RNA, DNA can also form G quadruplexes. This is a single, so 6-mer hexanucleotide, and immediately after addition of potassium chloride, it starts forming peaks in the region characteristic of G quadruplexes in the NMR. So when we leave this for some time, like here for one day, the peaks sharpen, indicating that there's a formation of a specific uniform structure. And then CD spectra of, so circular dichroism spectra of this shows a characteristic spectra that is characteristic of parallel G quadruplexes. We did this then for a 2-repeat G4C2, and what we see is basically that that immediately following potassium chloride, there's also some, so Watson Crick signal is here, so it's normal base pairing, and this is where the G quadruplex pairing comes in. So there is some formation, but it's a lot less uniform. And also, but there's still a lot of parallel structure happening in the 2-repeat. And then when we looked at the 4-repeat, so a lot, this is a lot of, mostly was, again, G quadruplex structure, a bit of Watson Crick, but maybe if you compare from here to, from the previous slide, what we have was a shift from the parallel repeats into the anti-parallel, or kind of a mixed repeat or mixed structure situation. So basically, getting back to the mechanism, the three possibilities: the haploinsufficiency, the repeat-associated RNA toxicity, and the RAN translation resulting in DPRs. We focused a lot of the work on the RAN toxicity, on the RNA toxicity, sorry. And the theory here is that we have the G4C2 RNA, which forms foci. It sequesters RNA-binding proteins that leads to their decrease for their normal function, and we have either neuronal cell death or leading to ALS and FTD. Now, we have drawn here as G quadruplexes binding these proteins, but that's not necessarily true. And I'll tell you one of the reasons why I think that later. So for our RNA pull-down, we went with a really long transcript. So we made a 48 repeat RNA, which we in vitro transcribed, and we added an S1 aptamer. So it acts like biotin. It will bind to streptavidin beads. And also for control, what we used sort of first 300 nucleotides of RFP, also bound to the S1 aptamer and S1 aptamer only. And the other thing that we have that we paid attention to was to incubate in a fresh nuclear cytoplasmic extracts. One of the things that I think a lot of you notice, basically, if you freeze the extract or cool it down, things start falling out, and I think it's a lot of that could be our proteins as well of interest. I mean, these are aggregation-prone proteins, so I think the importance of a fresh extract here is very very big, and I don't know, it could be some of the reasons why we have a lot of variability in different data from these RNA pull-downs. We looked at the nuclear and cytoplasm, as I said, and we looked at the cortex and cerebellum, and we saw several bands. Now, we're still following these up, so I really apologize for just labeling them as protein A or B. But we then looked at westerns so for following mass spec identification. We looked at westerns, and we saw really good validation of these pull downs. This is hnRNP-H, which basically, while we were doing this in Chris Shaw's lab, they were also looking for potential binders of the G4C2 repeat, and then we collaborated further on the hnRNP-H. I think nucleophosmin here is an important factor, because nucleophosmin is known to bind G quadruplexes, and we couldn't confirm it in cells. So one of my thoughts is that basically, in our pull downs, we do have G quadruplexes, so they're made, but then in cells, when we try to co-localize, it doesn't. So it's a kind of roundabout indication that G quadruplexes are not really happening in foci, in cells. In Chris Shaw's lab, they went about it differently. They looked at targeting screening. They did a targeted screening of hnRNPs and other RNA-binding proteins that might bind to GC stretches, and they had 3 hits which indicated that they could be bound to the RNA foci. So this is the RNA foci that were produced. So they had a 38 and a 72 repeat and an H repeat, and the foci formation are basically dependent on the repeat length. And it's been shown in, these foci have been shown to form in different cell types. So a little bit in sort of answering Aaron's question about this, whereas RAND translation and foci formation, one of the constructs for expressing cells was an EGFP construct with the G4C2 and the 3' UTR. So what happened was that the presence of the repeat reduced the amount of EGFP. So we had different within the same transfection, we had different populations of cells. We had cells which were expressing EGFP, but they had less foci, or cells that had foci, so they were in the nucleus, which meant that the RNA wasn't getting in the cytoplasm to have EGFP be translated. And this was translation dependent because PCRs of the messenger RNA showed no change in the amount of message depending on the length of the repeat. We have also synthesized another plasmid which we're now analyzing because G4C2 repeat is an intronic repeat. We have used these two vectors which have been shown in Bonanno et al. So it's basically EGFP or RFP which have an intron synthesized or inserted in the middle of the reading frame, and there is a small multiple clonic site which we expanded, and right now we have 128 repeat that basically shows RNA foci formation, and the cells that have the repeat are basically red because that means that it's spliced out. The intron remains in the nucleus, and the DS red is translated out in the cytoplasm. This is data from our paper where we show that in the screening where SC35, SF2, and hnRNP-H co-localize with the foci, and the strongest in terms of co-localization is hnRNP-H. We have done RNA pull-downs independently, so in Chris Shaw's lab and in my lab, and we have shown that in the pull-downs only hnRNP-H comes out and not SC35 and SF2. So they could have been a false positive out of the screen. Looking at the tissue in the cerebellum, we saw co-localizing, so mainly about 70% of the cells with foci had co-localization with the hnRNP-H. Then we looked at some of the, at the potential function of hnRNP-H, and there's a paper showing some effect of hnRNP-H loss or knockdown of hnRNP-H on splicing, and one of the targets is TARBP2, exon 7. So there could be reduction of exon 7 upon the knockdown of hnRNP-H, reduction of inclusion of exon 7 upon the knockdown of hnRNP-H. And we went to Ljubljana to look at this, and what we saw is that in the knockdown of hnRNP-H, we see basically this is 274 BP band is the inclusion of the exon, and 146 is the exclusion. So basically, with the knockdown, we have increase in the exclusion. There's also formation of this interesting 270 base pair band, which I will get into on the next slide. But also there's a decrease in the inclusion of the exon here, but I think that also goes towards because there's also this 270 band which appears. And we looked at that, and this is the region in TARBP2 where that is spliced out. So this is exon 7 that could be differentially spliced. And this is the region, and if you see some of the ESTs, there is actually an occurrence of a 4-nucleotide change of the in the splicing. So basically, there's an additional splice site within the intron, within the exon, and this splice site, actually, this region is relatively G-rich. There's 6 Gs in a region of 8 nucleotides. So the question here is just hypothetical. We don't have any data proving it. Does hnRNP-H bind here, or is the either knockdown or sequestration of hnRNP-H the cause of this 4-nucleotide reduction or expression of this exon that is 4 nucleotides shorter? So then the other two proteins A and B, we have looked at their co-localization with the repeats, and they do co-localize here shown in 72 repeats, and in the control there's no co-localization, and the same thing is happening with protein B, which we're now taking forward and looking at the postmortem tissue. So the work has been done, the RNA pull-down work and the G quadruplex work has been done in my lab in Slovenia. The RNA pull-down work has been done by Maja Staliker, a PhD student in my lab, and it's been taken over by Simona, the second PhD student in the lab. And the proteomics has been done in the center at the Jozef Stefan Institute, and we had help with NMR analysis at an adjacent institute in Slovenia, and it's been also in collaboration with Chris Shaw and Jörn Eola, and these are the people, the funding agencies. Thank you.